welcome back to my channel. Today the topic which we are going to deal with is combinatorial chemistry, right? Combinatorial chemistry involves what? Combinatorial synthesis, right? That means the synthesis of what? The synthesis of chemical compounds, right? Okay. And definitely, subsequently, what we should do with those compounds, right? Which are generated out of this combinatorial concept. Definitely, those compounds will be evaluated for their biological potential. So that means, basically, this is focusing, or the focus is on what? Generating a lead. Generating a lead. That means we are after developing or finding out what? The bioactive compound, right? Okay. We all know that in drug discovery, so many steps are there, right? There are several steps which are there in drug discovery, right? I just want to highlight few important steps, right? Why? Because we have to find out where exactly the combinatorial concept is applied, right? So first, we all know that a discovery process initiates with target identification, right? Definitely in this particular step, I don't think so that combinatorial chemistry is involved, right? Because this is the part which is attributed to the crystallographer, the NMR analyst, right? Or rather the biochemist, right? Okay, they will deal with this, the target identification, right? Because all those structures, all the structures of this macromolecule, they are stored in an effective way in the protein data bank, right? And how they are being stored out of what? Proper elucidation of what? The macromolecular structures. I'll not go into that particular topic, right? So in this case, we won't find any application of combinatorial chemistry. Let me do one thing. I'll just put an abbreviated term, or I'll just put the acronym of combinatorial chemistry like CC, or in short, Combicane, right? I can also write like Combicane. Right. Chalo. So this is the first step, right? The step with which the drug discovery initiates, right? The next is lead identification. Lead identification. Lead means the compound, though it is not having, the optimal biological effect, but still less than optimal biological effect that we can get out of this particular chemical entity. That is the lead. That means with which, upon optimization, we may land up to where? Land up to a drug molecule, right? Okay, chalo. So lead identification. In search of a lead, you have to generate what? You have to deal with so many compounds. And how those compounds, how the dealing operation will be there? It is out of what? Out of synthesizing, right? So it is out of synthesizing so many chemical compounds, you may land up to a particular compound which is found to be a lead, right? So that means the lead compound, the lead identification, right? So here the Combicane is applied, right? Okay. So once you got the lead, then what? You need to garnish the lead. Why? Because it is the lead from which you have got less than optimal biological effect. Now while garnishing, while decorating the lead, that means why you are going after decorating it? Just to ensure that with that decorated one, you are not supposed to get what? The one which you got from the lead. That means you are expecting even better, or rather I should say the best out of the best. Best out of best, right? Out of the best. So that means after decorating with certain substituents and all, we are optimizing the lead, right? And during this process of decoration, we have to deal with so many permutation and combination. So many insertions, so many removals, so many, changing of position of what? The substituents which you are going to incorporate in this particular lead, so that you can get a better, agent and all, right? Chalo. So that means subsequently we can say lead optimization, or in other words, the SAR, structure activity relationship, right? Chalo. So now another step which is there is lead optimization, or in other words, we are often calling it as SAR, right? Okay. So that means lead identification, lead optimization. These two are the steps of what? Drug discovery, where you can find the application of what? The combinatorial chemistry. These are the steps where combinatorial chemistry, the concept of combinatorial chemistry is incorporated. rather, or applied rather, right? Chalo. So with that we are done with what in drug discovery approach is it really needed to opt combinatorial chemistry? Okay, chalo. But you may raise a question that combinatorial chemistry, whether anything missing in case of conventional chemistry, because whether it is a combinatorial chemistry or certain other applied chemistry, you don't have to forget the conventional chemistry. That means the mechanism will remain same. The mechanism will remain same. The reagents are likely to be same, right? Then what is the basic difference between combinatorial chemistry and conventional chemistry, right? Conventional organic chemistry deals with what? The making of organic compounds and all, right? Combinatorial chemistry is also involved in making of the organic compounds. Then what is the difference? Where exactly they differ? So they differ in what? They differ in the variety. They differ in the number. They differ in the time, right? Okay. So they differ in what? They differ in time, number of compounds. So it is the conventional chemistry or conventional synthesis. Suppose we have a reagent called A, we have another reagent called B. What we are expecting when proper reaction will take place, then we are expecting what? The formation of a product called AB, right? Chalo. So this is about what? Conventional chemistry. Okay, so this is about what? Conventional chemistry, right? Okay, chalo. Conventional chemistry. Then what is combinatorial chemistry? In case of combinatorial chemistry, say okay, 1 to n, B also 1 to n. Suppose you have with you 5 components of A. What is that 5 components of A? Suppose you are in the hunt of making amides, right? You are in the hunt of making what? Amides. So what are amides? The principal functional unit that is present in the amide is found to be CONH. CONH, right? CONH, right? Okay, chalo. So that is, if we just split them all together, okay, we just split them from this area, we are going to get 2 different fragments, right? Upon completion of each fragments, definitely we can get the idea about the starting materials which are involved in making the amide, right? Okay, chalo. So in that case, that means you have to take what and how this merging will take place. That means what are the possible starting materials which are involved in making the amide. One may be an amine, the other may be an aryl halide or alkanoyl halide, right? Chalo, I'll just put here X. X can be chloro, X can be bromo, anything like, and here you have to put what? You have to insert one hydrogen to make it an amine, right? So when amine reacts with the derivative of what? Acid, carboxylic acid, right? Then we can expect there is a formation of what? Amide, right? Chalo. So this is the way amide will get formed. So what? Now the thing is that we are dealing with combinatorial concept, right? Here I have put what? A1 to n. That means this is the A which can give you 5. That means we are going to take 5 different or n number of components of A. So what does it mean? That means we have to deal with the analogues of A. Suppose A is found to be here the carboxylic acid derivative, right? So what do you mean by different components? Component means what? They must share a common substructure. Suppose we are dealing with what? Aryl acid, right? Chalo. So first aryl acid or halide I'll take, right? So different component means what? That means at different location of this particular acid, you are going to insert what? Substituent. So with respect to their substituent only they differ. So that is why I am telling what? They are found to be the component of A. So that means multiple components you can incorporate here. Suppose I am putting here Y. So in the next component I'm just shifting the Y from here to here. It has become a new entity, but they are sharing the common substructure. Got my point? Chalo. And same thing happens here also. Suppose I am going to take what? Aromatic amine. So I can change the what? I can change the pattern, I can change the feature of the aromatic ring system. In each and every location I can incorporate what are different type of substituents. So that means in what respect or with what respect they are found to be same, as they are sharing a common substructure. That means all are found to be amine. and all are found to be aromatic amines. Only thing is that they are substituted amines. So on the basis of the substituent, the presence of substituent or the pattern as well as the position of substituent, we can differentiate them. But they are originated from B. So that is why I am calling them as the components of B or components of A, right? So here what will happen? Here what will happen? Suppose ki I have taken the 5 components of A and 10 components of B. So what I can expect? I can expect 50 compounds. I can expect 50 compounds. So that means I am combining them all together. Okay? I am in the process of what? Combining them in every possible ways so that I can get 50 compounds. And if I run this particular reaction parallelly, that means I can save the time as well. In the process of making 50 compounds, if we stick to the conventional process, 50 days we have to wait. Why? Because 50 days means 50 times you have to repeat the operation, right? But here, as I have just, the setup is found to be parallel. So that means parallelly I am running this reaction with every components of A and B. And with that I can get what? 50 compounds at a time. So that means it is not found to be time taking, whereas the conventional one is found to be a time taking process. It's a time consuming process. So earlier if you stick to the conventional process, it takes around, it used to take around 15 years to generate a particular drug. Whereas through combinatorial chemistry, if you are just trying to develop a lead, it will take more than not more than 8 years, 5 to 8 years. Okay, enough for this combinatorial chemistry to generate a lead. But if we just want to get a lead from conventional one, it will you have to spend 40 years. Whereas in case of combinatorial chemistry, in a span of 8 years, you can land up to a lead, right? That is the advantage of this, okay? And why we are getting this advantage? The reason I have just explained, right? Chalo. Now, so we all know what is happening here, okay? What is happening here in combinatorial chemistry. Now, if we just try to figure out the class, if we just deal with the classification of combinatorial chemistry, right? The classification says that combinatorial synthesis, suppose ki I am just writing combinatorial synthesis. So one is found to be parallel. This can be broadly classified into 2 types: parallel combinatorial synthesis, and the other one is mixed. Mixed. It is the parallel. So what is that parallel? What parallel means? Parallel combinatorial synthesis. Parallel combinatorial synthesis means here also you are going to get n number of compounds in the same time. Okay. So that means the time consumption is found to be same in both the cases. But here in parallel synthesis, in each and every vessel, you will get a single product. Whereas in case of mixed, in a common vessel, you can get multiple products. Got my point? So here, in different vessels, you will get a single product, and here in the common vessel, you will get multiple products. Got my point? So this is the basic difference between parallel synthesis, parallel combinatorial synthesis, as well as mixed combinatorial synthesis, right? Chalo. Now let us deal with the requirements of combinatorial synthesis. Now let me tell you, and this is found to be one of the class, right? We have another class on the basis of the phase we are using, on the basis of the media that we are using, or the platform where exactly the compounds you are going to build. Okay, we can categorize them in 2 different parts, right? What are those? First one is your solid phase, and the second one is your solution phase. Solution phase, right? So, okay. So now this is found to be, so that means on the basis of the media that you are using or the phase you are using, they can be categorized into 2 different types. So one is found to be solid phase combinatorial synthesis, right? So here basically we are applying the first one, that means parallel synthesis, right? Parallel synthesis, right? But whereas in solution phase, there is every chance that in a single vessel you can get multiple products. The concept of mixed combinatorial synthesis, right? Where in a single vessel we are getting multiple products. But in solid phase, basically in a single phase, in a single bottle or single vessel, you can get a single product, or it may also be possible that in a common vessel you can get multiple products. That means both can be applied here. But in case of solution phase, mostly in a single vessel you can get multiple products. Got my point? Chalo. Now, so solid phase, solution phase. We'll see you one after another, right? Okay. So let us deal with the solid phase synthesis, solid phase combinatorial synthesis. So what do you mean phase, right? So phase means the support, right? The support. Phase is nothing but support. As I said, ki support, that means a platform where you are going to introduce what? Different reagents which you have designed or which you have incorporated while developing the scheme for that reaction, right? Okay, one after another you are bringing the reagents, right? And the reagents will interact all together so that finally you can get the product, right? Chalo. So in solid phase, phase means what? The support. The support is found to be an inert support is used, right? Inert support. Inert support. That means an inactive support. Usually they are made up of polymers, right? Usually they are made up of polymers, right? So support is here, the support we are calling is that resin bed. Resin bed. Now let me just chalk out that what are the, or let me just tell you that what are the basic requirements of solid phase combinatorial synthesis, right? So once we are done with the requirement, then we'll see how this can be operated and all, right? Okay. So the various requirements. So first and foremost, you need a support, right? So here we are going to deal with the requirements. Requirements of what? Solid phase parallel synthesis, right? Sure. Requirements. The first one is here, as I said, support, that is nothing but, or support material, that is nothing but resin bed, right? The next one is linker. I'll come to it while explaining each and every one, right? Okay, linkers. Then linkers with active functional group. Linkers with active functional groups. Then what next? The next is protecting groups. The third one, a mean to cleave the protecting groups, right? Or rather, a mean to cleave, a cleaving agent, I should say, because I'll just come to it. Cleaving agent, right? Cleaving agent, or we can say, and finally we need a deprotecting agent as well. Deprotecting agents, right? Okay. So with that we are done with the essential requirements of combinatorial chemistry or solid phase synthesis. First I have written here resin bed, right? We'll deal one after another, right? We'll deal one after another. We'll deal with them one after another. So first let us deal with the solid support and all, right? Okay. Now before that, let me just tell you one thing. I forgot to tell you. When combinatorial chemistry first came into the market, the concept came into the market as well as the application, right? Long back in the year of probably 1984, and after that, and now let me tell you, when in the year of 1984, combinatorial chemistry came into the market, right? Came into the discovery field, right? Before that, another technique was there, that is HTS, that is high throughput screening. That means you are screening means what? That means it's a bioassay, right? So you are just evaluating the quality of a particular compound, whether it is a bioactive one or an inactive one, right? So initially HTS came into the market, and later on it is the HTS that basically triggers the scientist to discover a mean by which they can feed the hungry beast. Why I'm telling? Am I deviating from my path? No. Is it a philosophical speculation? No. Is it a literature class? No. Then what? What I'm telling that it is the HTS. If we just consider the HTS is found to be a hungry beast, right? Why hungry beast? Because for a successful HTS run, you can evaluate the quality of a compound, say around 1,20,000 compounds. At a time, 1,20,000 compounds can be evaluated for their biological potential with the help of HTS. A successful HTS run involves 1,20,000 quality compounds. Got my point? And for that, the organization which are involved in that process, they have to bear 3 lakh USD. Got my point? So this is not that much easy, right? You must have some, you must be, financially sound so that you can have this HTS. So that means, now assume that the HTS, it can evaluate 1,20,000 compounds. Now, is it really possible to supply all this pool of compounds to this particular process simply with the help of conventional synthesis? No, it's not possible. That means we need to find some way by which we can at least hasten up the flow of making compounds and all, right? And that can only be done with this particular parallel synthesis, right? Okay, so that means it is the HTS being a hungry beast. You are just trying to feed the hungry beast with the enough number of compounds by violating the conventional one and making a new one, or rather I should not say violate, by altering the conventional one into a combinatorial one. Because we are not violating the basic principle of organic reaction. We are just alter the platform. We are just alter the platform of making the compounds and all. We are just altering the process through which we can generate diversified compounds, n number of compounds. Yes, mostly they are found to be same, but analogous way, they are found to be the analogs. They are found to be the analogs because we are altering them positionally, pattern wise. Not always pattern wise, sometimes position, chloro, sometimes ortho, sometimes meta, sometimes para, like this, right? Chalo. So that means, and all together, the combination, that means combinatorial chemistry, now it is combinatorial chemistry and HTS. If they are, they are working parallelly, or they are working in a friendly way, okay, they can fetch us a lead in a span of 5 to 8 years. This is the beauty of this hyphenated technique. Combinatorial chemistry provides the ample number of compounds which can be readily evaluated with the help of this HTS. So when this particular two techniques, they are functioning in a parallel way, in a subsequent way, you can readily develop or generate a lead or discover a lead in a span of 5 years. That means we can save a lot of times, right? Lot of time. Chalo. So just now I told about HTS and all, right? Now let us deal with this solid phase or support material, right? As I said, the support material is found to be most important, right? Support material. The support material is usually when the first, when it first came into the market, a scientist called Merrifield, right? Merrifield, okay. The scientist, he developed a particular resin which can serve as a bead, which can serve as a solid support, right? So it is after his name, the support material was found to be what? And that should be inert in nature. Inert in nature. It should not react with any of the reagents which are involved in the making of that particular compound, right? The compound which you are after, right? So that means the support material should not by any way react or alter the functional profile of n number of different starting materials or different reagents which are involved in the making of that particular compound, right? That means it should be an inert one. So in the Merrifield resin, the composition is found to be here basically polystyrenes, right? Okay, polystyrenes, they are cross-linked with 1% divinyl benzene, right? This was the combination, right? So here polystyrene actually, so here the composition is found to be styrene which is cross-linked with 1% divinyl benzene. So this is the composition of what? Merrifield. Merrifield resin, right? So this was the composition of Merrifield resin, that is styrene or polystyrene cross-linked with 1% divinyl benzene. Okay. Now the thing is that the problem was there. Some problem was there with this Merrifield resin because later on so many bids or so many resins came into the market. Why? Because there was some problem with the old resins, right? Okay. What was the problem? See, And when combinatorial chemistry was first used, or rather in its budding stage, okay, that time it was only restricted in making peptides. That means it was not meant for making non-peptides, organic molecules. Rather, it was only strictly, meant to develop peptides, n number of peptides, n number of combinations of peptides, right? So that means amino acids were there. Amino acids were there. So we know the nature of amino acids. They are found to be hydrophilic in nature, right? So what was the problem that they encountered by dealing with meriphyll resin? And soon after soon after identifying the problem associated with meriphyll resin, they developed another resin that is called, another support material that is called Shepard's polyamide resin. Shepard's polyamide resin. Polyamide resin, right? Okay. Polyamide. Okay. So what was the problem with styrene? Okay. What was the problem with this meriphyll resin? In meriphyll resin, actually, the support material was found to be hydrophobic, and the growing peptide was found to be hydrophilic. So at a certain point of time, when the reaction is approaching in the forward direction, there was a problem. That means it is out of hydrophobicity and hydrophilicity repulsion will take place. So all those hydrophilic amino acids at a particular point of time, they are just bending inwards, right? So bending on inwards, right? As they are bending on inwards, what happened? Hydrogen bonding, that means intramolecular hydrogen bonding took place. Intramolecular hydrogen bonding took place. It is because of the formation of hydrogen bond. What happens? Suppose you are in the process of making a nonapeptide, and this particular phenomena you observe when you are in the stage of pentapeptide. That means few more amino acids has to be incorporated. Now the problem is that as a result of this hydrogen bonding, what will happen? This will no more access any amino acid further. Why? Because hydrogen bonding has been taking place. There is no free amino group. There is no free C terminal. There is no free neither C terminal nor N terminal. So as a result of it, who will accept that the additional amino acid, right? So that means after a certain point of time, the reaction will be halted. The reaction will get ceased. This is found to be the limitation of what? Meriphyll resin, right? So later on, they move to Shepard's polyamide resin, right? Polyamide, what does it mean? Polyamide, that means subsequently what all the resins, what all the support materials they built, okay? They just kept in their mind that the resin must be an admixture of hydrophobicity and hydrophilicity, right? If the amalgamation is found to be proper, then we can at least overcome the problem which was there when we were in the meriphyll resin, right? When we people were using this meriphyll resin, right? Suppose. So polyamide means what? CONH. We should not say that CONH is found to be the hydrophobic one. It's a hydrophilic one, right? It is quite capable of solvate. It is quite capable of solvate others. So that means the support material itself is found to be partially hydrophilic. The growing peptide itself is hydrophilic. So with that we can avoid the chance of what? Chance of cessation. Chance of accidental cessation. Chance of accidental cessation of what? The forward reaction, right? Okay. And finally we can get our desired product. Okay. So this is about Shepard's polyamide resin. We have few more, this resin bits or the support materials. Okay, like your Tentagel resin. Okay, it is found to be much more, even more hydrophilic, and basically used for the making of what? Non-peptides as well. Because nowadays that limitation is not there, right? The limitation was initially it was only meant for what? Making peptides. But later on, as they modified the process, and suddenly they found the way by which they can make non-peptides, applying the concept of combinatorial chemistry, right? Okay. So chalo. So now the other one is found to be Tentagel resin. Here we are using 80% polyethylene glycol as well, right? Okay, Tentagel resin. So these are found to be the various support materials, okay? Support materials which they are using. Sometimes they are using glass bead as well. The derivatized glass bead as well as a support material, right? Okay.